Good afternoon and welcome to the e-Seminar Series 2022 of the European Society of Gene and Cell Therapy. My name is Hildegard Bühning. I'm a professor at Hannover Medical School and the current president of the ESGCT. The ESGCT is a non-profit organization promoting fundamental and clinical research in gene therapy, cell therapy, and vaccines. Providing training and education in these areas are part of our mission. We therefore launched the ESGCT Seminar Series. Today, we are already at the final seminar of our little seminar series entitled Viral versus Non-Viral Genetic Engineering of T Cells, a seminar series which was co-organized in collaboration with the American Society for Gene and Cell Therapy. Our final speaker is Professor Saul Priceman. He's an assistant professor and the associated director of translational science and technologies in the T-cell therapeutics research laboratories of City of Hope. He has received his BC in microbiology at the University of California, Santa Barbara, followed by a PhD in molecular and medical pharmacology at the University of California, Los Angeles. He is a trained tumor immunologist with expertise in T-cell biology and cancer immunology. We are now very much looking forward to his talk, which is entitled Advancing Immunotherapy Platforms for the Treatment of Solid Tumors.
Thank you so much for the introduction. I appreciate everybody who's in attendance live, and I apologize for my tardiness today. As was mentioned, my title is Advancing Immunotherapy Platforms for the Treatment of Solid Tumors. It's really around chimeric antigen receptor T-cell technology, which has taken the immunotherapy field by storm over the last 2 decades, certainly, but within the last 5 years, it really has taken shape. It's based on really conventional knowledge of how T-cells interact with a virally infected cell or a tumor cell. And it's through this TCR-MHC interaction, which in the bulk majority of initiating cancers, that might be effective at eliminating cancers. In the majority of advanced cancers, the mechanisms that are defective within that process can result in immune escape. And so nearly 20 or 25 years ago now, biologists identified a way to redirect T cells using a synthetic receptor that would identify a tumor antigen in the absence of MHC presentation. And this is chimeric antigen receptor technology. It's gone through multiple iterations, largely from first to second generation being the incorporation of a co-stimulatory domain on the intracellular side of the CAR that allows for greater persistence and cytotoxicity of CAR T cells. And then there are 3rd generation, 4th generation, and beyond that have incorporated other synthetic molecules that would allow for different activities within tumors. And really being taken by storm in the last 5 years due to the number of FDA approvals. We've had 5 now, 4 within CD19 directed CAR T cells for B-cell malignancies like ALL or B-cell lymphomas. And most recently, about a year ago now, with the FDA approval of BCMA CARs for patients with multiple myeloma. And because of the astounding therapeutic responses in hematologic malignancies, there are a number of groups around the nation and the globe that are redirecting CAR T cells for the treatment of solid tumors. You can see a list here that's now not exhaustive at all and about 2 years old, but really highlighting the number of targets, the number of disease indications, the iterations of CAR constructs, and the preconditioning, post-conditioning regimens. And yet the response rates in the comments section here are somewhat underwhelming as compared to hematologic malignancies. And so a number of resistance mechanisms have been identified, largely contributing to the lack of equivalent therapeutic responses in solid tumors as compared to hematologic malignancies. And I'm just highlighting 2 here because it's really driven what we're doing in my lab, but also driving what's happening in a number of labs in trying to improve on the efficacy of solid tumor-directed CAR T cells. Immunosuppressive tumor microenvironments, which are high macrophage content, low T cell content, and other variations in the tumor that create a hostile environment for T cells to survive and thrive and perform their anti-tumor functions. We're sort of overcoming those barriers using combinatorial therapies like radiation therapy, chemotherapy, and oncolytic viruses, for instance, and I'll show you some vignettes on what we're doing with that. There are intrinsic strategies, as I mentioned, 3rd generation, 4th generation CARs that are incorporating gene editing checkpoints, secreting immune checkpoint blocking antibodies, and targeting things like TGF-beta, which are highly expressed in an immunosuppressive microenvironment. We've also evaluated preconditioning regimens. If you're familiar with the CAR T cells in hematologic malignancies, lymphodepleting preconditioning regimens are required to unleash the activity of CD19 and other redirected CAR T cells. Also, routes of T cell administration can be used to incorporate T cells at the site of disease rather than just systemic administration, which most clinical development programs use. And the concept of repeat infusions to try and prolong the persistence, albeit artificially, of CAR T cells in a patient. There's also tumor antigen heterogeneity, especially in the advanced stage. Tumors are almost like mosaics with respect to antigens that are expressed on tumors. So single targeted CAR T cells may be unable to overcome that heterogeneity and would require multi-targeted CAR T cell strategies, as well as ways that you can spark an endogenous anti-tumor response, that it's beyond that of the single targeted CAR. This is just an illustration now, very old, but really highlights the manufacturing process, at least in autologous CAR T cell therapy, where patient A is leukapheresed, their T cells are harvested in the laboratory, they're engineered using various viral and non-viral engineering approaches to incorporate the CAR within the T cell. They're expanded 10 to 100-fold in cell number, oftentimes over 1 billion cells are frozen back, and then ultimately infused back into that same patient. And we highlighted various key variables that we think are still true today that would be required to manufacture the most optimal CAR T cell product. And in our program here at City of Hope, we've been going at it for just about 20 years now. Within the last 4 years, we've treated over 200 patients across a number of malignancies. You can see graphically depicted here. On the left, starting with CD19, as most programs have looked at in hematological malignancies, as well as in AML targeting CD123, and multiple myeloma targeting a different protein, not BCMA, but CS1. We've also treated a number of patients in glioblastoma. It really gets to the challenges facing solid tumor-directed CAR T cell therapies, and we've incorporated a novel route of administration to try and improve therapeutic responses for those patients. And with that experience, we also opened a trial now treating patients with brain metastatic disease. These are largely breast cancer patients that are HER2 positive, that have metastasis in the brain, where we're treating also locoregionally. We've also started a trial treating patients with prostate cancer. We've just received FDA clearance to start a trial in patients with advanced ovarian cancer, and so on, as our program grows. And just as our experience with treating locoregionally, IL-13 receptor alpha 2 directed CAR T cells, where IL-13 receptor alpha 2 is overexpressed in a number of glioblastoma patients and other primary brain tumors, we saw that local administration into the cavity that was remaining after surgical resection of the tumor allowed tumors to not reoccur quickly. And in a patient, actually in a case report we published several years ago now, with multifocal diseases, you could see here by MRI, and even a spinal metastasis, when we delivered CAR T cells not locally intracranially into the cavity, but rather intraventricularly into the CSF of the patient, you could see regression of all the multifocal lesions, suggesting that this regional administration could more could distribute across the central nervous system more effectively to target multiple lesions. And we've taken that similar approach and used mouse models to recapitulate orthotopic brain metastasis. This is a HER2 positive breast to brain metastasis patient-derived line we've orthotopically injected into mice, and evaluated two different CAR constructs targeting HER2. In this case, in this case, two different routes of administration, where we injected CAR T cells either locally into the brain tumor or IC, or intraventricularly or ICV. And you can see eradication of disease in all of the animals, all of the tumor-bearing animals, maybe a little bit slower of a response by treating regionally intraventricularly, likely because the T cells then have to traffic from the ventricle to the tumor. But what was really striking is if we compared intraventricular delivery regionally into the brain, into the CSF, to intravenous delivery, you can see dramatic responses by regional administration and only about a 50% curative response by intravenous administration, suggesting that locoregional administration of these CAR T cells is more effective than systemic delivery. Because of this, we've now started a trial treating patients with brain and or leptomeningeal metastasis by intraventricular delivery of these HER2 CAR T cells. The preclinical work was already published, and this was actually funded now by the California Institute for Regenerative Medicine, the CIRM agency. And so this trial is ongoing. The PI is Dr. Jana Portnow here at City of Hope, and we're treating in a single site phase 1 study. And our program has expanded in solid tumors to treat more than just brain tumors. We've gone after a target called prostate stem cell antigen that at the top here is expressed at very low levels in all normal tissue, maybe some higher levels in the stomach and bladder. And as compared to another target that's been very active in prostate cancer, being prostate-specific membrane antigen, which to our knowledge had higher normal tissue expression than did PSCA. And also PSCA is expressed at high levels in advanced disease and is correlated to negative prognostic values in patients with prostate cancer. And what we learned in building a more clinically relevant mouse model, in this case, and I think I'm showing data on the next slides, but really we evaluated preconditioning regimens because the field at the time didn't really appreciate that lymphodepleting chemotherapy could be required for solid tumor directed CAR T cells, while it was absolutely required for hematologic malignancies. And what we found was that preconditioning with cyclophosphamide, a lymphodepleting agent, could actually rewire the tumor microenvironment to allow T cells to infiltrate at higher levels and reverse immune suppression in many ways by shifting these M2 pro-tumor macrophages more to a pro-inflammatory macrophage that's less hostile to T cells, particularly adoptively transferred T cells. And by building this model, this really advanced beyond what we were doing in mouse models that lack an immune system. These are models that allow for human xenograft tumors and human CAR T cells to be injected, but really lack a competent immune system. And so to better understand safety and efficacy of our PSCA-directed CAR T cells for prostate cancer, we actually used a fully immune competent mouse model. We engineered mouse T cells to express a PSCA CAR that looked very similar to what we were putting forward clinically. And you can actually see here in this mouse, which is a knock-in using human PSCA gene under the mouse PSCA promoter, that there were several normal tissues that are complementary to human tissue that express low levels of PSCA, including the prostate, the normal prostate, bladder, and stomach. And so we could really evaluate both safety and efficacy. And what we found was striking as compared to our human tumor xenograft studies where there's no immune system, arguably. In this mouse model with PSCA CAR T cells, absolutely no therapeutic responses were seen, which kind of threw us for a loop several years ago. We didn't really understand exactly what was happening. But when we incorporated CPA or cyclophosphamide, which had its own anti-tumor response, but very transient, all the tumors came back, and combined now with the PSCA CAR T cells, we then uncovered about a 40 or 50% curative response in these animals, which suggested that preconditioning would be required for the therapeutic responses of PSCA CAR T cells. And in sort of understanding the mechanisms underlying this, we saw that cyclophosphamide actually improved T cell infiltration into the tumor bed, whereas otherwise the T cells were excluded and relegated to normal surrounding tissue. And when we combined with PSCA CAR T cells, we saw better early infiltration of the CAR T cells, as well as greater expansion over the first few days of those CAR T cells compared to mice that weren't preconditioned with this cyclophosphamide. And that was done by flux imaging of Firefly luciferase labeled CAR T cells prior to infusion. And when we looked at the tumor by RNA sequencing, we could see a shift to more immunologically warm tumors, higher pro-inflammatory cytokines, less M2-like macrophages, and greater antigen-presenting cell infiltration, supporting CAR T cells once infiltrating those tumors. And several years ago now, and we're wrapping these clinical studies up, we started a trial in patients with castration-resistant prostate cancer. The PI is Dr. Tanya Dorf, our own here at City of Hope, and we've been in a dose escalation and an incorporation of preconditioning lymphodepletion and doing several deep correlative studies to understand how patients are responding. And this trial is very exciting. We're coming near to the end of this trial and we'll present it soon. There are still major challenges. We know that the immunosuppressive tumor microenvironment can hamper overall responses of CAR T cells. And we've looked at ways to combine with other therapies. As I mentioned, radiation therapy has been shown both preclinically and clinically to warm up the immune microenvironment, release new antigens, spark an endogenous immune response. And a talented graduate student in my lab, Carrie Young, has looked at how radiation can do that in prostate cancer models. And you can see here, just as an example, there's greater infiltration of CD11C positive, MHC positive dendritic cells, a greater infiltration of endogenous T cells, and their greater activity is seen in tumors following radiation therapy. And when we combine it with our PSCA CAR T cells post preconditioning with cyclophosphamide, we see about 100% curative response now in this model, whereas before it was about a 40 to 50% curative response with CAR T cells alone, suggesting that this combination is a viable and good approach to improve the overall therapeutic response of CAR T cells. We've also looked more deeply at the immune microenvironment. We know in prostate cancer and other solid tumors that macrophages, and particularly M2 pro-tumor macrophages, are abundant, and they can potentially suppress CAR T cell activity. And in so studying this, we built an in vitro co-culture system that we could actually add various macrophage phenotypes to tumor cells and CAR T cells and understand the suppressive potential of these cells. And in building this model, this was also done by a scientist in the lab, Yuki Yamaguchi. We see that M1 macrophage co-culture with CAR T cells and tumor cells actually improves the CAR T cell killing capacity, whereas M2 macrophages suppress those T cells from killing, and that's shown here. We've actually seen that in both prostate cancer models, lymphoma models, as well as others. And in looking at mechanistically what's happening, we see that CAR T cells, which we know secrete a number of high levels of pro-inflammatory cytokines, can actually revert M2 macrophages to be more like M1 cells. This is shown just very simply by CD80 expression and CD163 expression, which are markers of M1 and M2 macrophages respectively. And we also see that adding atezolizumab, which is a PD-L1 blocking antibody, can reverse that suppression. And we've gone into more mechanistic understandings of how that can be, but suggesting again that in high macrophage-rich cancers, PD-L1 blocking antibodies may work in concert with CAR T cells to kill tumors. And sorry for the poor visual, at least on my screen. But we've also worked with collaborators. This is a collaborator of mine at Georgia Tech, Dr. Gabe Kwong, who is building heat-inducible gene switches in CAR T cells to try and regulate in a very inducible and spatially restricted manner any genes that they want to express in the CAR T cell. And we can modify the tumor microenvironment very selectively. In this case, we used HER2 CAR T cells that express a bispecific antibody, and only when it's heat conditioned in the tumor can you release that bispecific antibody. And we show that that combination can have very dramatic responses in terms of curative levels in a solid tumor model. Another challenge, which I mentioned before, is antigen selection, heterogeneity, and immune escape and tumor antigen escape following CAR T cell therapy. And in trying to get around this, we looked at other tumor-specific antigens that we could target in the microenvironment, which are not necessarily transcriptionally regulated, but looking at more post-translational modifications that are tumor selective. And in this case, aberrant glycosylations are known to be high in tumor cells versus normal cells. And we actually looked at tumor-associated glycoprotein 72, which has been around for quite a while. It's very cancer-specific, actually a pan-cancer marker, and expressed at very low to no levels in normal tissue. And in various cancers, including very high levels in pancreatic cancer and ovarian cancer, among others, you can see that TAG72 antigen expression is very highly expressed. As I mentioned, it's a pan-cancer target. It's been used in the past as theranostics for multiple cancer types. And actually, we came on after a phase 1 trial was performed in metastatic colon cancer with a TAG72 CAR, which was a first-generation CAR lacking a co-stimulatory domain, which we know now to be very poor in efficacy potential. But what this did highlight was safety and the potential for bioactivity. And so our mission really was to create a next generation TAG72 CAR that could offer benefits for patients with peritoneal ovarian cancer. And we know this because we looked at several fresh patient ovarian ascites from patients here at City of Hope, which had high levels of TAG72 expression, as well as other tumor types like patient-derived xenograft models of pancreatic cancer that also had very high levels of TAG72. And although this is published, and this was now done by a scientist in the lab who at the time was a graduate student, Dr. John Murad, who developed an intraperitoneal regional delivery model of TAG72 CAR T cells. This was the first iteration of a TAG72 CAR that had a co-stimulatory domain and was therefore deemed a second generation CAR. And you can see had dramatic early therapeutic activity in these animals. And though all tumors recurred in the animals, when we infused the CAR T cells, we prolonged the therapeutic response as long as the CAR T cells were repeatedly infused. And so it suggested that repeat intraperitoneal delivery of TAG72 CAR T cells could prolong survival in these ovarian peritoneal metastasis models. And what was striking is that early in the recurrences, and you can see this on panel A in the left, early in recurrences in the peritoneal cavity, these micrometastatic recurrences, they lacked TAG72 expression. But if you let those tumors grow for a bit longer, that TAG72 expression reemerged, which is suggesting that this post-translationally modified antigen is not necessarily susceptible to antigen escape as other transcriptionally regulated molecules are. And in fact, when we looked in vitro at just simple co-culture assays with TAG72 CARs, you can see downregulation of TAG72. And then when you take the tumors that are remaining, the residual tumors, and let them grow back out, you can see TAG72 expression reemerge. And that's in panel D. I'm sorry, I'm going to do a weird thing that allows my lights to go back on in my office. And so then we went on a campaign to optimize our TAG72 CAR T cell. And I'm just showing you 9 iterations of our TAG72 CAR, varying in the extracellular spacer domain, the transmembrane domain, as well as the co-stimulatory domain. We had already optimized the antigen binding domain, the SCFB for greatest activity. And this was done by a researcher in the lab, Eric Lee, who then went and looked at tumor cell killing in panel E, CD137 activation in F, PD1 as a marker of exhaustion in G, expansion in H, and cytokine production in I and J, being interferon gamma and IL-2. And although 3 or 4 constructs really showed pronounced functional activity as compared to the others, we looked at those 3 at greater lengths by using an intelligence assay that basically shows tumor growth kinetics by cell impedance. And you can see that one CAR iteration here had the greatest potent activity early and prolonged the anti-tumor response for over 10 days as compared to the others. And these 3 constructs only varied in the transmembrane domain and the co-stimulatory domain. When we went on to do a recursive re-challenge assay, which is adding tumor cells to the same T cells that are cultured in the dish, you can see again that last construct, the one with CD28 transmembrane domain and the 4-1BB co-stimulatory domain, showed the greatest tumor suppression, tumor killing activity, as well as the greatest T cell proliferative capacity over the re-challenge. And that was similar to what we found using a human xenograft model now, comparing those 3 constructs where we got 4 out of 10 or 40% curative responses with that CAR T cell as compared to 0 curative responses and recurrences in all the animals treated with the other 2 CARs. And you can see that we're still understanding the mechanism underlying this, but the CD28 transmembrane domain and 4-1BB co-stimulatory domain allowed for the greatest expansion of T cells regionally in the ascites that you can see in panel C, as well as an equivalent, at least, expansion in the peripheral blood as compared to another 4-1BB construct. And you can see that other groups have shown, and we showed here, that the CD28 co-stimulatory domain allowed for potent early activity, but couldn't maintain in persistence to overcome the tumor growth. And so this is actually, we've gained FDA approval at the end of 2021, and we're looking forward to treating patients in the next month or so with peritoneal ovarian cancer using this optimized TAG72 CAR T cell. But we've really gone on to looking at combinatorial immunotherapy strategies as a way to treat patients with multiple therapies that are safe, ideally that can create a better environment for CAR T cells to demonstrate their greatest potential. And in doing so, we looked at oncolytic viruses. These are really viruses that can selectively infect and replicate in tumor cells and not normal cells. They can cause direct tumor cell killing, and they promote immunogenic cell death like CAR T cells can, preconditioning can, and radiation can. This immunogenic cell death can induce endogenous immune responses that can work alone and in concert with other therapies. They also have the potential to be engineerable to express genes of interest that can be delivered selectively to tumors. And an example of that is the FDA-approved TVEC that is that Amgen developed, which is a herpes simplex virus 1 that's used to treat patients with advanced melanoma. But there are several challenges with using oncolytic viruses and other therapeutic agents as single therapies, and argue that combinatorial agents might work better. Our chief of surgery here at City of Hope, Dr. Yuman Fong, has been developing oncolytic viruses for over 2 decades now, and really landed on vaccinia virus as an oncolytic virus of choice. There are various attractive features to this, including broad tumor cell tropism, rapid replication. It's cytoplasmic without DNA integration, which can be potentially greater in safety than other oncolytic viruses. It's a large genome, which is excellent for introducing genes of interest. And this is just an example of what it looks like. Dr. Fong has done a lot of work in this area and actually has an ongoing clinical trial in a novel recombinant oncolytic virus he developed called CF33. And this is just an example of the potential in xenograft models of CF33. You can even see tremendous activity at the site of injection of that oncolytic virus where it's given intratumorally, but even in uninjected sites, you see anti-tumor responses and greater survival in mice bearing multiple tumors. He's gone on to show that this CF33 vaccinia virus has tremendous anti-tumor activity in a number of tumor models, including pancreas, colorectal, and lung. And he's gone on to actually start a clinical trial in patients with triple-negative breast cancer with this CF33. And together we collaborated and came up with an idea that was really simple in nature and performed by a talented graduate student, now scientist in my lab, Anthony Park, which was the following. If an oncolytic virus can infect tumor cells specifically, why can't we engineer that oncolytic virus to express a gene of interest? In this case, CD19, that could shuttle to the surface of an infected cell, be expressed on that surface, and then we could use CD19 CAR T cells, which are FDA approved now for multiple hematologic malignancies, that could be redirected to a solid tumor, any solid tumor, in a tumor agnostic manner, and target in combination those tumor cells, and have the added benefit that oncolytic viruses have, which is that they can warm up the microenvironment, reverse some of the immune suppression, and have potent tumor responses in combination with CARs. And we went on a campaign to do this. We've published this now about a year ago, but we developed a CF33 vaccinia that expressed a human CD19 gene that was actually truncated on the intracellular domain that lacked signaling capacity. But what it still did is present the antigen that CD19 CAR T cells can recognize. And you can see pretty pictures here in purple showing expression of CD19 on a triple-negative breast cancer, for instance, following infection with this vaccinia virus. And what was really striking is this, what we call therapeutic window of opportunity, where we infected tumor cells with CF33 CD19T. You can see dramatic CD19 expression at very low multiplicity of infection of this virus. And a lot of these cells, up to 60% of these cells, were still viable, suggesting that now we could come in with a combination of CD19 CAR T cells and mop up the remaining cells that express CD19 but are not dead. And in doing so in vitro, we showed dramatic therapeutic responses in combination of this oncolytic virus and CD19 CAR T cells. And there are some images here and quantification, but really showing that there is this synergistic killing potential where at very low MOIs, you see a suboptimal killing with the oncolytic virus alone in black and gray bars, but in blue in combination with CD19, now we're CD19 CAR T cells, we're now killing many more tumor cells in the dish. We showed this actually in xenograft models where the combination generated eradication of tumor cells, whereas the individual agents alone had less of a response. And we built, again, a fully immune competent mouse model so we could understand how this combination works, what mechanisms are underlying the therapeutic response. And again, you can see the oncolytic virus has very potent infiltrating, immunological responses by infiltrating T cells, endogenous T cells, into tumors. And the combination of CD19 CAR T cells with the oncolytic virus dramatically improved that further. We looked at the tumors by RNA sequencing, and you can again see dramatic immune responses that are happening by the single and the combination agent. And the virus actually improves CD19 CAR T cell trafficking to the tumors. We showed this again by non-invasive flux imaging of CD19 CAR T cells. And what was perhaps most striking is that in tumors that were cured by the combination, when we re-challenged those animals with tumor cells, and these are wild-type tumor cells not expressing CD19 and have never been in contact with the virus, those failed to grow in these animals, suggesting that there was protective anti-tumor immunity ensuing following the combinatorial. And another sort of unpredicted benefit of the combination is virus release. We know that once a virus infects a cell, kills that cell, it can release a number of particles to neighboring cells and infect. And we see that in green here by the virus alone and virus plus mock unengineered T cells. But when we combined it with CD19 CAR T cells, we could see a significant enhancement of viral spread within the tumor. This was quantified by immunofluorescence staining as well as flow cytometry, and it was very tumor specific. So if we looked at other tissues, including spleen, liver, lung, and ovaries, you didn't see that infection, and you didn't see an enhancement by the combination with CD19 CAR T cells. And so in sum, where do we go from here? We're continuing to learn lessons from our phase 1 trials. We have ongoing prostate cancer, brain metastasis, soon to be initiated ovarian cancer and pancreatic cancer trials. And we're learning more at the bench to bedside level how to overcome these challenges in solid tumors, being tumor antigen escape and immune suppression, and really trying to understand what combinatorial agents may best improve the therapeutic responses of CAR T cells and provide durable anti-tumor responses for patients. And so I'd like to acknowledge this now old picture of the laboratory. Several folks are not seen here, but you can see their names. And I mentioned them for each of the projects. They've really done a fantastic job in the lab. I work within a fantastic program that is putting forward translational CAR T cell programs, and various collaborators I work with and funding agencies who have allowed this to happen. And I thank you. And if you're live, I'll take any questions.
Thank you very much for this fantastic talk and for sharing all these details. I wanted to put some questions to you, and the first one, I would like to come back to your CAR redoing or tailoring. So are there any tips you do have for the audience where in particular to focus on? You looked more into the signal domain, but from your experience.
You know, even the traditional non-signaling transmembrane domain we've shown has a great impact on the CAR, even above and beyond the stability of the CAR expression on the surface that has been shown with the transmembrane domain. I think the biggest key for CAR T cells is the specificity of the antigen binding domain. That is a critical component. There is no solid tumor antigen that's like CD19, for instance. CD19 is really restricted to a cell type, B cells, and is expressed on both normal B cells and tumor B cells, and you can live with B cell aplasia. But for instance, there are several targets, including HER2, that could potentially create havoc in terms of normal cell targeting and toxicities if the CAR T cell is not optimized. We've optimized our HER2 CAR T cell. We know that collaborators at Baylor School of Medicine have optimized their HER2 CAR T cell, but it's on the heels of some pretty bad toxicities that were shown at the NCI with a HER2 CAR T cell about 10 years ago. So we're all cautious about targets that we go after, and we've done a tremendous amount of work to try and optimize the CAR for greatest specificity.
I mean, therefore, I also very much appreciated and liked your work on the new antigens, such as the glucosylation, and really see and dive deeper into really finding differences and really improving specificity. Yeah. When we come to your nice last part where you showed us the results on the oncolytic viruses, can you just allude a bit on the timing? So when should you do what?
Yeah, it's been the greatest challenge for us. You know, in theory, the virus needs to be there, infect tumor cells, and express a large level of CD19 for the combination to work most effectively. But as it turns out, CAR T cells take a while to get to tumors. Their activity is somewhat slow compared to, for instance, cytotoxic agents and other things. And so I really think this has been the challenge in developing the clinical trial. You know, I'm fortunate to work with a lot of amazing scientists and clinicians. For instance, Yuman Fong, who has a great amount of experience in developing and clinically developing oncolytic viruses and other immunotherapies. So we are building out the design of the clinical trial, I think, will be limited because of what the FDA will deem as potentially safe. But also we'll hope to try some variations in the timing, as you mentioned, because I think that will be an absolutely critical component to elucidating the greatest therapeutic potential of this combination.
I mean, and it maybe gets even more complicated when we go back to your first part of the talk, where you also showed the impact of preconditioning combination then between cyclophosphamide and radiation, and so on and so forth. So if you can step back a minute and look, what would be, from the perspective you are at, the results you have at the moment, what would be the best way to go when you would like to create an effective treatment for your CAR T cells for solid tumors?
We've really only touched the surface of how preconditioning regimens can impact solid tumors. We're stealing basically from active therapeutic agents that have been looked at in hematologic malignancies. I mean, arguably, cyclophosphamide has no business in treating prostate cancer patients. It is active in ovarian cancer, so we're sort of intrigued on the potential of this preconditioning for patients with ovarian cancer. Fludarabine is a therapeutic agent that's used almost exclusively for heme malignancies. Again, probably not really active in the large majority of solid tumors. So a lot of our work is actually looking at preconditioning, not only in the agents that we use, but what their impact is specifically on the microenvironment, in addition to what they do in lymphodepleting or creating space for CAR T cells to set up shop in lymphoid tissue. And so we're looking at other agents. And as we talked about with the oncolytic virus, timing is also critical. Prior to doing the cyclophosphamide studies in the animals, for instance, we did a time course to understand the lymphodepleting activities and the impact on the microenvironment specifically in animals, because we really had no idea the kinetics of either, and we didn't know if one was required for the other. Did you need to lymphodeplete the mouse to get the greatest improvement in the microenvironment? It turns out that they're probably a little bit exclusive, and we don't know if that's really going on in patients. So a lot of our work is looking at biopsy material and blood and other tissues to try and understand the kinetics of CAR T cells, but also what we have to go with in terms of the preconditioning regimen that we're using.
Thank you. And so I don't have any further questions. I don't see any further questions. So I would like again to thank you for your really wonderful talk. And I would like to close the session here and say goodbye and thanks again.
Thank you again.